はい、皆さんおはようございます。日本の皆さんおはようございます。そしてですね、サンディエゴをはじめとする米国の皆さんこんばんは。LINK-Jのですね、ライブウェビナーにご参加をいただきまして、誠にありがとうございます。私、LINK-Jの曽山と申しまして、今日のですね、全体の司会、そして冒頭のご挨拶をやらせていただきます。今日はですね、生命科学分野のイノベーションを実現するデジタルテクノロジー、デジタルテクノロジー for Life Science Innovation 第4回 LINK-J UC サンディエゴライフサイエンスシンポジウムという、テーマがちょっと長いんですけれども、こちらのシンポジウムを、お送りいたします。今日はですね、画面でお示ししてますね、こちらのプログラムに沿って進めてまいります。ハイブリッドで開催ですね、リアルおよびですね、オンラインということで、会場はですね、日本橋ライフサイエンスハブというところで行っておりまして、こちらの会場にいらっしゃる方はもちろんですが、オンラインでご参加いただいている方もですね、Q&A 機能、こちらから、ぜひね、ご質問をいただければ、幸いです。Zoom からのご質問の方はですね、セッション中、から受け付けておりますので、どなたへのご質問かということを、冒頭にですね、書いていただいた上でご質問を書いていただけると、大変助かります。またですね、こちらの会場、でご参加の方のみとはなりますが、講演終了後にネットワーキングの時間も用意しておりますのでですね、ぜひご登壇者の皆様、また、参加者同士でですね、皆様で、交流を深めていただければというふうに思います。またですね、このイベント、同時通訳およびですね、ウェビナーの英語字幕サービスを使っております。英語字幕の方はですね、市販アプリを使用しておりますので、その内容ですね、語訳とか訳文の欠落、等、ある可能性がございますが、そちらに関してはですね、LINK-J、および UC サンディエゴ校は、責任を負いかねますので、この点はご了承を願います。さらにですね、本ウェビナーの録画、そして録音、画面キャプチャー、こちらに関してはですね、恐縮ですが、固くお断りをしておりますので、ご了承ください。最後になりますが、ウェビナー終了後にですね、表示されるリンクよりですね、ぜひアンケート、ございますので、こちら皆さんのご意見、ご感想をお聞かせいただければ幸いです。今後のですね、セミナーと、皆さんのお役に立てるようにですね、やっていきたいと思っております。というところで、ここまでがハウスキーピングイシュー的なところなんですが、今日はですね、UC サンディエゴ校様とものライブウェビナーになってます。UC サンディエゴ校の皆様とはですね、LINK-Jはとってもですね、助けていただいております。LINK-J 設立されたのが今から6年ちょっと前の2016年の3月なんですが、UC サンディエゴ校とですね、その2ヶ月後にですね、コラボレーションの覚書 MOU を結んで、それ以来活発にですね、日本およびサンディエゴ等でですね、コラボレーションを、やらせていただいてます。コロナの前はですね、大体、年に1回、UC サンディエゴ校の、著名なですね、世界的に著名な研究者の方を日本にお招きして、特別、セミナーを企画しておりましたし、コロナ後、のこの2年間ぐらいはですね、両社でですね、オンラインセミナーをですね、ここ1年半とか2年の間に、12回ほど開催をしております。そして今日はですね、UC サンディエゴ校のですね、キーパーソン中のキーパーソン、のお三方が来日されるということで、この来日に合わせてのシンポジウムの開催となっております。まずお一人目はですね、ジェイコブススクールオブエンジニアリング、UC サンディエゴ校で最大、確か学生の数がですね、皆さん、ファカルティも合わせて1万人、そしてファカルティの方の数でも300人以上という工学部のですね、学部長でいらっしゃるアルバート・ピサノ学部長、そしてお二人目はですね、同じく、工学部の中にあるんですが、インスティテュート・フォー・グローバル・アントレプレナー、こちらのエグゼクティブ・ディレクターをなさっているデニス・アブレムスキーさん、そして、全学 UC サンディエゴ校全学のですね、インターナショナルアウトリーチのディレクター、オフィス・オブ・リサーチ・アフェアスですね、こちらのですね、和賀美和子さん、このお三方のご来日、そして今日ご参加をいただくということでございます。それでは和賀さん、よろしくお願いします。
曽山さん、ご紹介ありがとうございます。 皆様おはようございます。私は UC サンディエゴで国際アウトリーチのディレクターを務めております、和賀でございます。本日は進行役を務めますので、どうぞよろしくお願いいたします。さて、UC サンディエゴに新たに完成した工学研究教育棟のフランクリン・アントニーホールは、デジタル未来へのコラボラトリーを10室以上収容しています。コラボラトリーとは様々な専門分野の教授陣、学生、業界パートナーが共通の目標に向かって協調的に研究を進めるための開放的な大型研究室です。本日のシンポジウムではまずピサの工学部長、研究課長よりこの新しい工学研究棟の狙いと概要を説明し、続いて生命科学分野に関連する2人の専門家がプレシジョンジノミックスとエンジニアードナチュラルインテリジェンスについて技術講演を行います。また UC サンディエゴの戦略研究連携先である大阪大学からも特別講演をいただきます。技術講演の後にはパネルディスカッション形式で海外進出を目指す生命技術系生命科学系スタートアップにとって国際的なアクセラレーションプログラムがどのような価値を持つかを UC サンディエゴの IGE 並びに早稲田大学、筑波大学でスタートアップ育成に携わっている専門家に語っていただきます。このセッションでは実例としてそれぞれの大学のスタートアップを短い動画でご紹介します。それでは最初のスピーカーをご紹介します。Now let me introduce the first speaker. Dr. Aldo Pisano is the Dean of the Jacob School of Engineering at UC San Diego. He assumed the current position in 2013. Prior to that, he was a faculty member at UC Berkeley for nearly 30 years. He's a member of the U.S. National Academy of Engineering and is the founding director of the Dean's Roundtable sponsored by NAE. Dean Pisano, the floor is yours.
Okay, I think I'm on. Can you hear me? Oh, sorry. Okay, good. Okay, please forgive technical difficulties. I'm jet lagged. So, good morning, everyone here in Japan. Good afternoon, everyone in California and in the United States. I'm very happy to make this presentation here to you today. So and I thank Mr. Toyama and Ms. Waga to make a very nice introduction. The name of my talk is, how do we build and strengthen high-impact innovation ecosystems in UC San Diego? And perhaps there are some lessons that might be good for Japan and for other places. And in return, I'm always looking for better ideas. So if I made a mistake, please point it out, and I'll be happy to correct it and change our ways so that we can be very, very effective. So when I became the dean 8 years ago, I realized that the engineering school needed to be closely connected to every other school on campus. And so the story I am telling is an 8-year-old story, 8 years of hard work in which first we formed intense connections to the entire campus. And so, for example, if I look at the lower right corner, you will see the Jacob School of Engineering is tightly connected to the School of Physical Sciences and Biological Sciences through a special institute called the Institute for materials discovery and design. I will not read the entire view graph. The point of the view graph is to show that sustained innovation requires the construction of many, many pipelines and many, many connections, because you never know which one you will need when the big new idea arrives. And so this picture shows that first, the engineering school found 10 other parts of campus and made 10 very strong connections to those parts. The second step in the plan was to allow the faculty to practice working together as a unified group. Now, this did not come completely naturally to the School of Engineering. When I arrived, there were a few very active centers, but not so many. Now, I know that pairs of faculty were already collaborating, but they did not form formal relationships. They did not form a special new entity called a center in order to have a particular charter. And so the next step was to launch a program called the Agile Research Centers, and then connect them all to industry. So I will not read all the names for the sake of time, but I will point out that there are Agile centers such as machine intelligence, computing and security. And if you think of a future of digital health, is that not important? Or perhaps wearable sensors, would that not be important? Or contextual robotics, robots that understand the context or the meaning of the situation, would that not be important? And you can imagine that after these centers are formed and anywhere from 3 to 8 faculty are working together, you might get very exciting results in each of these individual areas. And indeed, for the past 7 years, that's what we've been doing, building these centers. Now, we know the centers are successful. I will not read all the names on this view graph, and I'm sorry, it looks more like an eye test. Can you read the tiny print? The point is, these centers created these connections to these industry partners, just from these centers alone. So we added some almost 80 new connections to the school just from allowing faculty to practice working together on projects that have industry relevance. So you can see momentum building, right? The faculty work together better, the industry is working with faculty better, and the faculty are working with their colleagues across the campus better. We have not yet spoken about innovation. We're just talking about doing the homework, building the foundation. The next piece was the corporate affiliates program, which is a special program that companies have to pay a fee in order to get specialized student talent placed in the companies. For the sake of time, I will just point out one special feature. The corporate affiliates pay an annual fee. There are approximately 75 to 80 of them every year. When a company is looking for talent, they tell us what is the topic, what kind of talent, and how many. We never tell the companies to search a database. We handpick 10 students for every slot the company requests. So if Google says, I want to hire 3 programmers, we send them a list of 30. My school has 9,400 students. I would never send a list of 9,400 students to industry. But a list of 30 is manageable. including their resumes, by the way. So the next piece is to convince the companies that not just the research, but the talent can be had. So what is the digital future? The digital future is our vision for using technology, creating technology, to assist people in their pursuit of health and happiness. The research vision is to look at new data streams, combine them with real-time analytics, make sure we have cybersecurity, and then provide actions or agents. An agent is software that does something for you. An action is advice you're given so you can do it yourself. And we support all of this on a commercialization platform, which of course is our own IGE, the Institute for the Global Entrepreneur. So up to now, I've described all the homework we did over the last 8 years to get ready for a big innovation ecosystem. Now, by ecosystem, I mean an active, responding, alive, changing system of people working together. They change how they work based on the circumstances. And this ecosystem will be moved into one new large building, 17,000 square meters, 13 special collaboratories called Franklin Antonio Hall. This is a picture of one floor of the building. The front doors of the 4 big rooms all are within 10 meters of each other. So imagine a front door in each corner of this room, and they all have to mingle on the way to the elevator or the staircase. There were 3 rules given the architect. The building must, naturally, by the way it's designed, maximize the circulation of people and ideas, provide flexible collaboratories that need not be renovated. And our interpretation of that is no fixed walls, everything's on wheels. And finally, welcome industry, because if we don't have industry partners, we do not have a sustainable ecosystem. So for example, what new projects can I start very rapidly now that I have the faculty talking to each other, the Agile centers all working, the collaboratories all filled, and the industries all engaged? How do I use this momentum? So for example, I can quickly start an innovation ecosystem in autonomous transportation, advanced topics in self-driving cars and trucks, for example, or boats, or aircraft. And just looking at the names of the collaboratories that are already in the building. Global Entrepreneur for startups, integrated computing and security for the brains of the autonomous vehicle. The Data Science Institute, because we have to teach the vehicle to drive. Wearable Sensors, where the vehicle wears the sensors. Contextual robotics, the self-driving car is a robot, of course. Sustainable power and energy, super high capacity batteries, and advances in wireless communication. Of course, autonomous vehicles need to communicate with each other and with the environment. Or, more related to today's topic, how might we put together a digital health innovation ecosystem? Well, starting from the left, machine intelligence, computing and security, of course, entrepreneurship, of course, systems engineering, yes, wearable sensors, of course, maybe microbiome, or in the upper right, nano-immuno engineering or precision genomics. So the collaboratories are all large, they all have special projects of their own, but because they are close and they must mingle, now our next step is to have all these collaboratories work together. So for the sake of time, I will not read about each collaboratory. We have 2 speakers from 2 different collaboratories today. And I thank Vinit Bafna and Geert Kouwenbergs for their efforts to put together their presentations. Let me just page through. I'm submitting this material just for the files so that if this is recorded, someone can go back and ask me questions. And I will just say that the plan is that the sum is greater, the entity is greater than the sum of the parts. The purpose is that individual successes in collaboratories become enhanced successes for the ecosystem, where the ecosystem is a vibrant, changing, organic group of people who are modifying their work and research goals month by month in order to have the impact for the digital future. So I thank you very, very much for your time and attention. And this is my last view graph.
Thank you. Thank you, Dean Pisano. We have one question from the audience, and you have covered to some extent. The question is, what is the core competence of this department, he said. However, I think it's the Franklin Antonio Hall. Would you rephrase the core competence, please?
The core competence is the disciplines required to create digital technology combined with 5 or 6 new applications. So you may note that this is not a biomedical research building. It is a digital future, digital technology for health or for a human happiness building. And so therefore, you can imagine everything will have electronics and wireless and special materials in it, but then we will add all the other topics from health and medicine. So it's the fusion of engineering and health sciences.
Thank you very much. Our next speaker is Dr. Vinit Bafna, Professor of Computer Science and Engineering in the UC San Diego Jacobs School of Engineering. He joined the computer science faculty in 2003 after 7 years in biosciences industry. He has co-authored 150 research articles in the leading journals in the field. In 2019, he was selected as a fellow of the International Society of Computational Biology. Today, he will talk about the Center for Precision Genomics and, in particular, complex structural rearrangements in cancer. Dr. Bafna, please take the microphone.
Thank you. Let me share my screen and I can start. Hopefully everybody can hear me and can see my screen. If I speak too fast, please feel free to interrupt me at any time. And you can also ask me questions at any time. I have no problems answering before my talk is over. I'd like to start by first thanking Ms. Vaga, Mr. Somaya, and the other organizers for inviting me to speak today. We have just started this collaboratory on which is the Center for Precision Medicine. And I would like to start by talking a little bit about our collaboratory, and then I'll end with a small research vignette of some of the research that we are currently working on. So I think it's well appreciated now that we are in the post-genomic era, where genomes of many individuals can be completely sequenced. And we are not that far away from when the first genome was sequenced. The draft genome was only sequenced in 2001, and it was a project that I participated in many years ago. But the situation has changed in the last 20 years. Technologies have advanced to the point where all of us will have our genome sequenced in our lifetimes, perhaps many times over. And the mission of our center is really to analyze this vast wealth of information from genomes and to enable scientific discoveries that directly impact healthcare. Specifically, we will use techniques from algorithms, from data science, statistics, and also our knowledge of genetics and biology in order to extract where the variation in DNA lies and what is the consequence of this variation on human health and disease. So we are a group of 5 core faculty members, and I'll introduce them in a minute. And I would like to say that all of us have already had a career of interdisciplinary research, and we have also had many collaborations. If you look at the slide here, some of these small circles indicate the existing collaborations that are within the among the faculty members of this collaboratory. But you can also see that we were quite spread apart. So some of us were in computer science and engineering, in electrical engineering, in bioengineering, but also in departments that are both physically and in terms of the scope of research quite far away. So we have colleagues from pediatrics, from pharmacy, from medicine, and from the Data Science Institute. So we are very excited because we are finally going to be co-located in a facility which will have both wet labs and as well as computational resources, and we can truly collaborate at a scale that was not possible before. Right. So Dean Pisano has already shown us what the map looks like, but I thought I might also share this with you. So we have a collaboratory that has room for 8 PIs, and currently there are 5 of us, but we hope to recruit more faculty in the future. And we also have space for a wet lab right in the middle, and it's surrounded by computational scientists and their workstations. And so we are very excited. I think we hope to move within the month or so, and our collaboratory will be fully active at that time. So, all right, so let me also introduce the team members. First, Melissa Jimrik. She's an assistant professor who has a joint appointment in medicine and in computer science and engineering. And she's the lead for the center. She couldn't be here today, and so I'm going to describe the center in her absence. And her research is very interesting. She also looks at DNA variation, but instead of looking at small mutations. So here you can see the 4 nucleotides that comprise of the DNA, and you can see it in this position. Some individuals have a T in the position and other individuals have an A. These are small nucleotide or single nucleotide changes, or SNPs for short, and they are the most widely studied mutations. But what Melissa studies are mutations that are of a different form, where you have nucleotides that are repeated many times. And so one individual may have 5 copies of these repeats. Another individual might have 6 copies, and so on. And the variation in the number of copies of these short and tandem repeat elements can have severe phenotypic consequences and can result in many different genetic disorders, as well as other kinds of variation. Another colleague is Professor Sheng Zhong. He has a broad research agenda, but currently he's looking at early diagnosis of many diseases such as Alzheimer's disease. And specifically, they are an RNA lab. So they study also nucleotides, but ribonucleotides, nucleotide acids, and they are looking at RNA elements that have come out from the cell, and they are in the extracellular domain and can be studied and form markers for some of these diseases, allowing us to detect these diseases many years before they manifest themselves. My colleague, Siavash Mirara, is an evolutionary biologist, and his expertise is in looking at genomes of thousands of individuals from a species or from multiple species and recreating their evolutionary trees, including, for example, transmission networks of HIV, metagenomic analyses, and also looking at genome skims where a very small amount of sequence is collected from an individual for very low expense, and that data can be mined very quickly to identify what that unknown species might be. Alon Goren looks at what we call epigenetic phenomenon, and here we think about the DNA as being tightly wrapped around proteins called histones, and the modifications in these proteins, as well as the unraveling of this DNA when it's tightly bound or when it's loose, can have a lot of impact on the genes that are on the DNA, the expression of the genes and their ability to make proteins. And so by controlling the expression, we can actually both sort of identify disorders, but also find therapeutic interventions for them. And then finally, my own research is on genomics. We look at many different sort of variations in genomes, specifically large variations where big chunks of the DNA can break apart, they can circularize, they can invert, and they can rearrange in myriad ways. And specifically, we are looking at extrachromosomal DNA in cancer, and I will spend some time talking about it today. And I also look, along with Siavash Mirara, we are also looking at genome skimming as a technique for quickly identifying species in an ecological environment. So just to give some examples, actually, let me see if there is a question on the chat box. Right, so one of our attendees asks, what is the relation in application with artificial intelligence and genomic editing? And I will give you some examples of that in a few slides, but I'll also have time to answer questions afterwards. But thank you for the question. So one of the research projects from the Jimrik lab, where they looked at expansions of short tandem repeats and found thousands of genes whose expression was really associated with the number of copies of these short tandem repeats. This was a study published a few years ago in Nature Genetics, and they found that STRs can actually modify the expression of genes involved in many complex diseases, including schizophrenia, inflammatory bowel disease, and even common phenotypes such as height and intelligence. Dr. Jimrik, Melissa Jimrik, with other colleagues in the School of Medicine, have now been funded for a Center for Admixture Science and Technology. And the idea here is that each of us as an individuals, we are the mix of many, many ethnicities because of recent admixture. For example, sort of African migration into North America We have many African American ethnicities available, and their response, their expression of the genes in these individuals can be quite different. And what they are studying is specifically individuals that are recently admixed, but whose genomes are not well studied. They are not well represented in the large-scale genetic studies that are being done. And this center seeks to remove this inequity by focusing on those individuals and trying to identify the genetic basis of disorders in these individuals. And towards that research, we also collaborated to identify slightly longer tandem repeats. We call these variable number tandem repeats, which mediate the expression of proximal genes. And this way we could use them to detect phenotypes. And one of the attendees had asked a question about the use of artificial intelligence. So in this project specifically that's listed out here, we looked at neural networks, not deep neural networks, but with sort of shallow neural networks to quickly identify regions of the genome that encode these variable number tandem repeats. And once we have done this quick identification, then we use other sort of machine learning methods, such as hidden Markov models, to be able to precisely count the number of these repeat elements, because the repeat elements are inexact and they are difficult to count without having this kind of generative model. In Professor Mirarab's research, they recently created a phylogeny of more than 10,000 species, and that revealed some very interesting proximity or similarity between two very distinct domains of bacteria and archaea. And this research was recently published in Nature Communications. And then with Mirarab, we have been working, as I mentioned, on using genome skims for sample identification. In Professor Zong's research, they just recently used extracellular RNA in order to be able to detect biomarkers that can predict individuals who are more likely to develop Alzheimer's disease in a few years' time. This is, of course, very preliminary data. It doesn't have clinical utility yet, but it definitely points to the need for future studies that can validate these markers as true biomarkers for predicting of the disease. All right, so in the short time that I have left, I'd like to explore a little bit of my own research in detail, which is talking about complex structural rearrangements in cancer. And specifically, I'd like to talk about extrachromosomal DNA, which have been known for a long time. In fact, the first discovery of these elements was back in 1965, when people looking at cancer genomes. So here we are looking at cells when the chromosomes, when the cells are in metaphase and the chromosomes are very tightly compacted. So you can actually seek out individual chromosomes in the cell. But you find that in addition to the chromosomes in these cancer cells, we also see these small elements which are extrachromosomal. And it turned out that these extrachromosomal elements do not have functioning centromeres. So when the cells divide, they can randomly get a partition into the two daughter cells. And this unequal division followed by selection can lead to tremendous growth in copy number of these extrachromosomal elements, causing a lot of pathogenic conditions. Right? And so very recently, of course, we used some electron microscopy in collaboration with some collaborators here in the Cancer Institute. And you can see very clearly that these elements are both different from the chromosome, extrachromosomal, as well as they are circular. And we can also see these elements replicating and forming copies of themselves. Right? So the question that we really set out to ask was how prevalent, how common are these extrachromosomal DNA elements? And we started on that research many years ago now, about 7 years ago, where we looked at 2,000 cellular images in metaphase. And so you can see on the top right, these are images where the DNA is stained with DAPI, and you can, and it's compact in metaphase, so you can quickly identify the extrachromosomal DNA elements. And we used a UNet, a specific type of deep neural network, to be able to analyze these images directly and count the extrachromosomal DNA elements, allowing us to look at thousands of samples and actually documenting how common these are in cancer samples. And the results were quite surprising, because the previous best analysis suggested that only 1.5% of all individuals with cancer had ecDNA, but our results suggested that almost 40% of the individuals with cancer could have extrachromosomal DNA. Right? I'm going to take a quick break to just see if there's any other question. No, there isn't. And I'm actually answering the questions live, but I suppose I'm supposed to type the answer, and I will do that momentarily. All right, so you can see out here that we have different tumor types and different prevalences, and certainly certain brain cancers have a very high incidence of extrachromosomal DNA, but others do not. But overall, almost 30 to 40% of the individuals have cancer. So this was, of course, data where we could only use cell lines, and we couldn't make use of this kind of information in primary tumors because we don't have growing cells which make this kind of image analysis possible. And so we developed some genomic tools. Specifically, we developed a tool called Amplicon Architect that looks at the genomes identified from the individuals with cancer. We map the genomes back to a reference framework and look at these discordant mappings where portions of the genome map very far away when they should be mapping together, suggesting that they have rearranged in the tumor genome. And we use this information to make a graph, sort of a discrete graph, and then trace cycles in this graph to be able to identify extrachromosomal DNA elements, as well as identify the exact genomic structure of these extrachromosomal DNA elements. Right? And we tested the ability of these cycles to be able to predict ecDNA. And we find that as the number of ecDNA per cell grows in the images, our predictions are almost perfect out here. So we can predict these extrachromosomal DNA elements with over 80% precision and 80% recall. And this makes it a good tool to actually look at primary tumor samples and identify presence or absence of extrachromosomal DNA. And so we look at the same similar data as I had shown before, but this time in 2,000 individuals, primary tumor cancer samples, individuals with cancer. And again, we see a huge variety of individuals and cancers that carry ecDNA, and we find it in close to 30% of all the individuals. On the other hand, when we look at the DNA from the matched normal region of the same individuals, normal tissue and normal blood, we do not see any incidence of this cancer, suggesting that they are uniquely found in cancer. So our research since that time has been at finding out properties of this cancer. And many other researchers have now got into the game, and this has become a very important area of research. And so one of the things that we find is that genes that are on this are very active. They make many copies of the protein. And if these genes are oncogenic, that is, they indicate proliferation, they will quickly make the cancer very pathogenic. And the increase in the expression of these genes is more than what could be explained by copy number, suggesting that something is very different about these extrachromosomal DNA. Moreover, individuals that carry these extrachromosomal DNA have very poor survival relative to other individuals that do not carry these ecDNA. So clearly they indicate the most pathogenic forms of cancers, and targeting these extrachromosomal DNA would be a very important thing. And in fact, I'm associated with a company that is developing therapies to actually target these extrachromosomal DNA. We are also looking at many other forms of genomic technologies, including technologies that say which portions of the DNA elements are proximal. And we find when we study with these that new proximities are created due to extrachromosomal DNA formation. So if there are elements very far away from the gene that could not actually regulate the gene, when they are on extrachromosomal DNA, those enhancer elements are hijacked and they can be used to regulate the gene, causing a very different sort of genetics and a different phenotype that's produced by having these extrachromosomal DNA. Moreover, these extrachromosomal DNA are mobile and they move around and they hit other chromosomes. And in hitting those chromosomes, they may regulate genes on the chromosomes. And therefore, they can cause a tremendous change in the entire gene expression profile of that individual. So because of the high interest of this, the Cancer Research UK and the National Cancer Institute have now established very large awards. And our team is one of the teams competing for one of these $25 million awards. And the goal of this challenge is to understand the biology of ecDNA generation and also to understand their origin, their evolution, genomic organization, and to identify therapeutic vulnerabilities that will allow us to target and find targets for intractable cancers. All right, so with this, I would like to thank you for listening. And we, of course, invite you to this beautiful space that has that's available to us at UC San Diego. We invite you to visit and collaborate with us. Thank you.
Dr. Bafna, thank you very much. There is one more question. So can you tell us how the collaboration between 2 or 3 faculty members can also result in benefiting the collaboratory as a whole?
Yes. So, so one of the things is that many of us have independent collaborations with many scientists in the medical school. But because our core members have a certain core set of expertise in genomics, in computational sciences, in machine learning, actually our collaborations are greatly enriched with discussion with our core collaboratory members. But that is not easy when we are so far apart. So, for example, Siavash Mirarab, a colleague of mine, we would like to study evolution of these extrachromosomal DNA and build the phylogenies within the tumor itself. But these kinds of discussions are difficult to have because we are currently in different buildings. And while we do meet quite often, I think having, meeting in one location would be much more conducive. Another big benefit of the collaboratory is that it's the first time that many of us are co-located with a wet lab. And so, for example, one of the things that we are studying is specific genes where increase in the number of tandem repeats can be actually causal for a disease. These are very hard to study with whole genomes because it's very expensive to generate long reads that are required to study. And so our collaborators in the collaboratory now, we are working to identify these genes and actually do targeted sequencing specifically of these genes of interest. And in that experiment, we would have very sort of low throughput, but very targeted sequencing. And the computational people could analyze all of that data in real time, allowing for the next set of experiments to be done. And so that will really speed up the cycle of experiment and computation that we would like to achieve.
Thank you very much. Okay, so let's move on to the next speaker. Thank you, Vinit. UC San Diego has a strategic research partnership with Osaka University. For this symposium, we have invited Professor Hideo Matsuda in the Department of Bioinformatic Engineering at the Graduate School of Information Science and Technology. His research interests include genomic data analysis, gene regulatory networks, and gene expression analysis. He has a collaboration with Dr. Bafna. Professor Matsuda will present a collaborative study on tracking cell migration in live animals in Japanese. 松田先生、お願いします。
はい、ありがとうございます。大阪大学の松田と申します。はい、ちょっと、今日いる前に、まず最初に、このような機会にお招きいただきました、LINK-J 並びに、我が様をはじめとする UC サンディエゴの関係者の皆様方に感謝いたします。それでは、講演を始めさせていただきます。見えてますでしょうか。はい。はい。それでは始めさせていただきます。まずこういう、セルマイグレーション、有層細胞の、追跡の研究でございます。で、まず背景の説明をさせていただきたいと思います。で、この研究は、細胞の追跡、画像からの細胞の追跡なんですが、追跡の対象となるのは白血球ですね。ここに上がってる、これが白血球です。ホワイトブラッドセルと書いてますけど、あるいは専門用語でルーコサイトと言われたりします。で、これが、外からこうバクテリアが侵入してきたり、あるいはウイルスが侵入してきたりすると、いろいろ動き回って、それに対する、防御をするということです。で、普段こういう白血球は、血管とか、いろんなところにいるんですけども、それがこうやってこれが血管の細胞なんですけど、血管の中にこう入り込んでダーッとこう進んでいくような非常に複雑な動きをします。ちなみにこれは赤血球ですね。血管の中にいる細胞です。で、これをどう追跡するかなんですけども、まず研究体制をご紹介したいと思います。大阪大学の医学部の、免疫細胞生物学教室の石井勝先生の研究室と、私の研究室で、共同研究をしております。で、石井先生からは顕微鏡の、イメージデータを提供いただいて、それに対する、この細胞の動きの、解析を、画像処理で行うというものでした。で、当初はこの細胞の追跡ということに関してあまり我々詳しくないというか、始めたばかりだったので、いろんなやり方を試しておりました。で、そこで、先ほど講演をしていただいたバフナ先生、UC サンディエゴのバフナ先生のグループが、先ほどエキストラクロモゾーマル DNA、核外のDNA ですけども、それを画像解析で、検出するという研究で、非常にそういう画像処理に関する、詳しい知見をお持ちということで、そこの後で出てきます、バフナン先生の研究室の大学院生が、オブジェクトトラッキング、を、深層学習を元に、物体追跡ですね。そういう共同研究を提案していただいて、一緒に研究を始めたということでございます。で、実際に細胞の動いている、白血球の動いている様子がここに示されています。で、これ、3枚ありますのは、 それぞれ別々の刺激剤、リポポリサッカロイド、リポーターとか、いろんな、違う種類の刺激剤で、見ていただくと、なんとなく動きが違うということがわかります。で、こういうデータを、画像データを、しかもこれ動画像ですね。ライブセル、生きた細胞の、動きを、画像として提供していただいて、で、今これお見せしているのは、すでに、既存の使われている、リンエージトラッカーという、そういう細胞追跡ツールです。で、追跡できているのはなんとなくわかると思うんですけど、時々すぐ途切れてしまうということがあります。で、なんとなくこれ見ていると、わかる範囲で、既存のツールでわかる範囲でなんですけども、すごく短い間隔でくるくる回っているような動きと、あと直進しているストレートな動きとっていうのが混在しているような、そういう形を印象を受けるということですね。これをもうちょっと詳しく解析したいということなんですが、ただ、このトラッキングのその精度の限界が、既存のこのリンエージトラッカーにはありまして、でもっと精度の高い追跡が必要であるというふうになりました。で、それに対して、先ほどのバフナ先生のスライドにも出てきたんですけども、ラジクマーさん、これ、バフナ研究室のその博士課程の、大学院生なんですけども、この、大学院生は、非常に優秀な人で、こういう、コンピュータービジョンという分野で、こういう物体追跡の、そういうコンテストがあるんですけど、そこで非常に優秀な成績を、収めた、MD ネット、マルチドメインコンボリューショナルニューラルネットワークというのを、使うというのを提案していただいて、で、これを細胞の追跡に使ってみようと。で、このためには、教師データがトレーニングデータが必要なんですけども、そういう正解データは、石井先生の研究室で、1251個の、細胞のこういう軌跡に関して、提供いただきました。で、その結果がこちらでして、で、これ左が先ほどお見せしたリンエージトラッカーという既存の細胞追跡で、で、こちらは、そのラジクマーさんが、開発された追跡ツールですね。で、こっち見ていただくと、これ全く同じ画像なんですが、すぐ途切れてしまうということでして、すぐ軌跡が消えちゃって、ここで違う色になったということは違う細胞として認識されたということになります。で、こちらは、この緑が正解、本当に人間目で、判定した正解で、赤がこの MD ネットというツールで、追跡した結果です。で、もう非常にちゃんと、追跡がこう、きっちりできているということがわかると思います。で、こういう形で、かなり追跡ができるようになってきたんですが、まだ若干十分でないところがありまして、時々細胞を、の追跡が、取り違えというか別の細胞の方に、間違って、いってしまうということがあります。後で例をお見せします。で、これを解決するために、これなんでそもそもそういう追跡ミスが起こりかっていうと、これ実際に細胞は3次元の空間で動いてるもんですから、そこで、なんですか、重なってしまう、上から見てるような形で、その3次元的な動きが、上の方から見た画像だと、重なってしまうときがあると。だから、深さ方向の動きをちゃんと考えないといけない。で、あと、いろいろトレーニングデータを増やさせた方が精度が上がるということで、ちょっとこの数値でお見せしてるんですが、ラジクマさんの、ものだと、こっちが追跡した結果が正しかったら 100% で、それの割合なんですけど、78.7%、結構いいんですけど、時々、追跡が、ミスが起こる。で、こっちは、本来追跡しなきゃいけない正解をどれくらい取りこぼしなく追跡できたかということで、大体8割弱の精度でした。で、これに対して、ちなみに先ほど、ちょっと条件が違うんですけども、既存のツールだとなんか3割ぐらいだったと。で、これに対して若干のこういう深さ方向の、改良を加えたことによって、これだけ精度が向上したということです。で、ちょっと例をお見せしたいんですけども、で、左がオリジナルの、もので、右がこういう深さ方向の解析を、検討したものです。で、見ていただくと、このタイミングで実は重なってしまって、で、元の方だともう重なってしまうと、細胞はどっちがどっちか分からなくなってしまって、ここからミスが起こっちゃうんですけど、当たり前っては当たり前なんですけど、この深さ方向の、これちょっと画面には見えてないんですけど、こういう深さ方向の情報まできちっと入れると、その追跡を、この場合はちゃんと連続して行うことができるというものでございます。ちなみにこれ私の研究室の修士課程の水垣君っていう学生が、改良してくれたというものです。で、さらにですね、もっとちょっと頑張らないでないかということで、私の研究室の別の、学生、博士の学生の、藤本君っていう学生が、もっとさらに、もうちょっと新しい、物体追跡の、やり方を、応用してくれました。で、ちなみにこれその時の物体追跡のツールが使ってるコンテストの画像なんですけど、ちなみにこういうコンピュータービジョンの物体追跡ってこういうふうに、よく使われるのが、こういう、一般のこういう都市の中の人の歩行者の動きですね。これを追跡するというものです。はい。で、これを、使うんですけど、単純に使うだけだとそんなに性能が変わらないので、この藤本君は、ビデオフレームのインターポレーション、保管をしてみようということです。で、どういうことかと申しますと、これでちょっと見ていただければわかるかと思うんですが、この左側がオリジナルの顕微鏡画像、顕微鏡動画像ですね。で、右側が保管した画像となっています。で、なんか、あんまり違いがないように見えるんですが、よく見ると、左側は、すごくこう、なんていうか、コマ落としというか、なんかカクカクしたような動きになっているかと思います。で、右側はそれに対して、なんか、じわーっという、割と、滑らかな動きに見えると思います。で、これなぜかっていうとですね、ちょっと今まで全然説明してなかったんですけど、この、生体内で細胞を、きちっとこう、追跡するために、顕微鏡で、見るときに、これ、特殊な、レーザーを当てるような特殊な顕微鏡を使ってまして、で、非常に強い光を当てるんですね。で、そうすると、ずっと、非常に短い時間間隔で、フレームを、すごく、早い時間間隔でいっぱいレーザーを当てると、細胞がすごくこう、ダメージを受けて、で、そこでちょっと、動きが止まってしまうわけですね。要するに、細胞の動きが、ちゃんと追えないと。で、そのために、実際にはこれ、ちょっとわかりにくいんですけど、1分間隔で、だから実際の画像はもっともっと、1分間隔で、カクというような動きになってしまっています。で、そうすると1分の間に、だいぶ細胞が動いてしまって、で、そこでこう、うまく追跡できないということが起こります。で、これに対して、画像保管、フレーム保管の技術っていうのがあるというのを藤本君が見つけまして、で、要するにこの1分間隔で撮った画像の間を、対応づけて、こう、要は間の画像を、生成するということです。で、 こちらが、それで、その保管を行ったもので、15秒間隔の画像。だから、1分の間じゃなくて、さらにその間を、間に3つ、画像を入れたような、そういう形になっています。で、そうすると非常にこう滑らかに、細胞が動くような画像が得られるので、追跡がだいぶやりやすくなるということです。で、実際にちょっとこの、先ほどのような細胞追跡を、この、保管をさらに使って、追跡を行ったのが、この画像でございます。で、ちょっと、実際にはすごくたくさんの、細胞を追跡しないといけないっていうのと、で、これでも、たんまにちょっとミスるんですけども、かなりの精度で追跡ができているというのが、見て取れたかと思われます。はい。で、はい。で、以上のように、バフナー先生のグループで最初、その、物体追跡を細胞の、動きの追跡に、適用していただいたっていうのを、叩き台にして、バフナー先生のグループと私のグループで、共同研究で、白血球の郵送の追跡というのを、深層学習をベースに、作ったということです。で、元画像は、医学部の石井先生から提供いただいて、で、これ、実際に活躍したのは、それぞれの研究室の、大学院生の人たちで、バフナー先生の研究室のラジクマンさんとか、私の研究室の水垣君、藤本君が、共同研究で、お互い大学院生で、それぞれこう、なんていうか、本当に共同で、交流して、実際のこういう、研究成果を上げたということになります。で、協調で、国際会議で、それぞれ発表したということでございます。こういう共同研究が、そういう、コンピューターサイエンスの、物体追跡の研究が、こういう細胞の、追跡で、役に立つという、そういうのを共同研究で行ったということでございました。ご清聴ありがとうございました。
松田先生、ありがとうございます。ちょっと日本語で質問一点させていただきたいんですけれども、
はい、
学生さん同士でこうコラボレーションされたかと思うんですが、
はい、
外国の、パートナーの、機関の人が来ることで、その日本の学生さんたちにとってどのような影響がありましたでしょうか。もしよろしければ一言お願いいたします。
はい。やはり当初、こういう経験は私も初めてだったので、本当にうまく共同研究できるんだろうかということが非常に懸念だったんですけども、実はどちらも大学院生、身分が近くて、年も近くて、で、やってる研究も結構近かったということで、本当に思うよりもすごくスムーズに学生同士で交流をしたということです。で、最近はこういうソフトウェアの共同開発をするためのGitHub とかいろんな環境が整備されてますので、そういうのを使って、言葉の壁はあるんですけども、実際に動くプログラムを相互に共有して、いろいろやり取りをしたということです。ちょっとお答えになってますでしょうか。はい。
ありがとうございます。年代も近いし、コミュニケーションも取りやすかったということですね。
そうですね。
はい。またぜひコラボラトリーの方にもお越しくださいますようにお待ちしております。Thank you very much. Thank you very much. Let's move on to the next speaker.
ありがとうございました。
So our next speaker is Dr. Gerd Cohenberg, who is a professor of bioengineering in the Jacobs School of Engineering and co-director of the Institute for Neural Computation at UC San Diego. His research focuses on neuromorphic engineering, adaptive intelligent systems, neuron silicon and brain-machine interfaces, and micropower biomedical instrumentation. Dr. Kohlenberg will discuss engineered natural intelligence, adaptive neural systems in silicon. Dr. Kohlenberg, please take the mic.
Thank you so much, and it's my pleasure to be here and present for you and interact with the audience here. This will be about engineered natural intelligence and the work we're doing in this area at the Jacobs School of Engineering, and we very much welcome interactions with our partners or colleagues in Japan. And so hopefully this will spur some interactions, some collaborations in which we can further advance this technology that we're developing here for the digital future. And please interrupt me for any questions any time during my talk. So let me first say what natural or this engineered natural intelligence is about. And perhaps this could be seen as a misnomer, because typically natural intelligence is something in nature and engineering is typically thought of being artificial. But the whole point here is that we take inspiration from biology, from neuroscience, in building silicon engineered microchips that inherit some of the same principles of efficiency, resilience, this natural intelligence that we have in biology. And this way we're shaping the digital future. At the same time, we use then silicon integrated circuits, custom integrated circuits, to also interface directly with biology, neurobiology, at scale in order to probe function and modulate function, which allows them to observe neural systems, natural neural systems, as they interact naturally with their environments. And that way we can then construct better models of systems interacting also more naturally with their environments. And then this is really a way of shaping the digital future, which is what we're about in the Franklin Antonio Hall. And so one of these centers that we have now at Jacobs School of Engineering, as we heard from Dean Pisano, is then this Center for Engineered Natural Intelligence, along with many other centers that also embrace this digital future with life sciences innovation. And so the specific objectives here, as I just showed, right? So we have engineering, we have neuroscience and the environment. And so we want to create this very efficient interface between them. So CENI, the Center for Engineered Natural Intelligence, the faculty are then attempting to have better understanding for how the brain works as an engineered system, and then interface to the brain through technology, and use all that knowledge then towards new applications, engineering and machine learning for the digital future. And so we're so fortunate then to have, thanks to the benefactor Franklin Antonio, to have this new environment where we can have this ecosystem of all these interactions between engineers, neuroscientists, clinicians, that's right, to all come together and do this really study how the brain works and really make inroads in applying all the knowledge for engineered systems. And so we can then visualize brain activity in real time with our new wireless dry electrode, electroencephalogram, and functional near-infrared spectroscopy systems, really observing the brain at new unprecedented scale in mobile systems, in mobile environments. We have this great infrastructure for a massive data access with 100 gigabit Ethernet really wired in this collaborators, directly connected to the supercomputer center that we have also on campus. And then, but the really exciting thing is the interaction that we can have. And so those hubs allow them to get collaborators from industry, from academia, to join us. And so we have welcomed the partners in industry to be part of these collaborators and work with us closely. And so they're very welcome. So we also extend this welcome here to our Japanese colleagues here and partners. And so as Dean Pisano was mentioning, so we get this really interesting ecosystem through this very interactive, this setting, right, where people really talk to each other. They have to collaborate because they just have joint spaces where they do so. And so then we have those interactions with this CENI, this Engineering Natural Intelligence, with other centers, such as the Center for Wearable Sensors, which also then goes towards a digital future, but in this case, by then interfacing to the body at scale with sensors that can directly probe and modulate function. And this, of course, is also very important to understand how the brain works in mobile settings. And so we have many interactions with that center. In addition to other centers, such as the Center for Wireless Communications, yet another example of the synergies that we have at this interface between engineering, neuroscience, and medicine. So I'll give a few examples then of those synergies and where our collaborations with multiple faculty working together are then really advancing all those interfaces towards the digital future. And so one very important one that was started 30 years ago, but is now really becoming at the forefront thanks to our efforts, is in neuromorphic engineering, which takes inspiration from the structure, but also the organization, the function, as well as the structural organization, and the fundamental physical principles of how brains operate. And then have this and morph that into silicon systems that inherit some of the same structure and same principles. Basically building circuits, literally building circuits, emulating directly the physical substrates that we have in biology. And this way, those systems then inherit the natural intelligence of their counterparts in biology, as well as their extreme efficiency and resilience. And this is very important for maintaining sustainable future. We really want to cut down on the power consumption of our computational systems, and so efficiency is extremely important, as well as resilience. We also want to make sure those systems are then very resilient against even imprecisions that they're inherent due to the extreme small size of this circuits for extreme efficiency, right? So, and we still want these circuits to operate correctly. So and so we have this great inspiration from how the brain does that at multiple levels of scale, all the way from whole brain dynamics down to the scale of individual ions moving to ion channels. And neuroscientists have really made great inroads in understanding how the brain works at the fundamental level through this analysis approach, which is really a top-down approach, right? So studying function really at the elementary level, down to this levels of synapses and ion channels. And so great advances have been made, but this is really a top-down approach, and typically in a very detailed fashion, which then loses out on some of the higher level representations and putting everything together. So, but then engineering or systems engineering is the counterpart of that, basically synthesizing, basically then able to synthesize brains from the ground up as a bottom-up approach, right? Basically constructing models of the brain, in this case, at each level of representation, and then basically have those models being abstracting just at each level of representation, just what is needed to understand just what is happening at that level. And this gives you great efficiency in representation for really building systems from the ground up. And this allows them to really study then the brain at scale, right? And so it just so happens that at each level of this representation, down from the ion channels all the way up to whole brain level, there are this isomorphism, there's this parallelisms between how the physics operates in biology versus how the same physics operates in silicon integrated systems. For instance, with ion channels, right? The same Boltzmann physics of how electron, Boltzmann physics of how the ions move to ion channels also play out in silicon, right? Where you have electrons and holes also moving with similar exponential distribution of current versus voltage in those semiconductor channels, right, of transistors. And so neuromorphic engineers have taken advantage of the similarities of really building then brains from the ground up that really adhere to all those isomorphisms in giving you rise to extreme efficiency and resilience in the representation. And so I'll just give some examples of that. So here is, so we have been building silicon retinas, right? Basically models of the visual system, including the optic nerve in silicon, where you're not just taking pixels of information that is just basically a transmitted pixel at a time, but rather than you can do some local processing, in this case for decoding motion locally in the pixel, and then encoding that efficiency. So only a pixel that sees any change in intensity is encoded as an on or off event, just the same way as the retina and the optic nerve will communicate those on and off events very efficiently. And the efficiency gives you the rise to extreme, algorithmic efficiency of how to then extract information from the environment and actually do some computation locally, in this case, locally in the focal plane. And so then taking this one step further, so then we have chips that the silicon integrated circuits that will do something with those events, those on and off events that are created by those imager or some other sensors, silicon copias, there's other examples. And then those neuromorphic, the synaptic arrays will then process those events, those on and off events, and then do some inference, so basically detecting some objects or doing some other decision making. And so there we have extreme efficiency in those crossbar arrays of synapses that basically implement connections between neurons that model basically how the brain does the information processing at extreme efficiency, where in those crossbars, right, will just interconnect neurons vertically and horizontally at great efficiency. In this case, our circuits here are able to operate basically at just a few femtojoules of energy per synaptic operation, right? So for each spike that a neuron sends, basically each synapse here consumes only about 13 femtojoules per synaptic operation. And that's actually comparable to the human brain, which consumes about 1 femtojoule of energy per synaptic operation. And so we're getting close to the efficiency of doing the processing in those silicon integrated circuits. And this particular architecture also then allows us to do this reconfigurable connectivity where we can go forward and backward in this across this array, which allows us to do backpropagation learning or efficient autoencoders. This restricted Boltzmann machine or autoencoders for very efficient generative as well as discriminative training and learning, all basically efficiently done in silicon. So here's an example of how this top-down and bottom-up, this inference and generation, this going both directions, this reconfigurable forward and backward connectivity to the array can give rise to great efficiency in this representations, in this case, for doing variational autoencoding, right, alternating between inference and generation very efficiently, for basically filling in the blanks of missing information that you have. In this case, so this generative models allow to not only detect and recognize what is happening, but also filling in missing information and just basically reconstruct noisy samples that you have. And that's a very important aspect of natural intelligence. So here's a demonstration of how that is happening in almost real time. This is a demo that's been recorded of this system, this chip, this silicon integrated circuit operating. So here you have an image of a 6, and then you perturb here the pixels, you include some pixels and change them. And so it's now a noisy 6 that is now then detected, but also reconstructed, right, by this hardware happening in real time. And so they can compare the energy for doing this, or this chip, against what the what software is doing, and it's going to be this orders of magnitude better. So in this case, we just have a few nanojoules of energy for doing this entire MNIST type of a detection as well as reconstruction. And so I can just play it again here, right? So you perturb a few pixels, so it becomes less of a 6 visually, but still, as you say, recover, the system will recover it, and then it can also then give you so he detects a 6 and also then recovers it. And so we can take this to great levels of scale. So we have now developed systems all the way now to 116 million neurons and 40 billion synapses that we're now catering to the community at large for basically developments in neuromorphic computing. And so this is now being hosted. It is basically an array of field programmable gate arrays that we developed. And so we're now hosting this at a workshop on neuromorphic engineering this summer in Telluride, where the community can then engage with this hardware that we developed. And so similar to what we did last year, right? So we build this hardware that also then helps the community to try out neuromorphic approaches. And so this is a means for the neuromorphic engineering community, but also the neurosciences community, the computational neuroscience community, to try out those advances and really then connect them with artificial intelligence or natural intelligence at scale. And so we also take this efficiency to extremes as well. So here we have work where those crossbar arrays of synapses can also operate at really extreme efficiencies, owing to basically recycling even the energy that it takes for driving those memory elements that basically do this computing in memory. At extreme efficiencies, basically we even couple the capacitive energy that is consumed, well, that is circulated in the array, by coupling all that energy externally to an inductor, a coil, basically circulating them between electrical and magnetic energies, recycling the energy. In this way, we operate at energy efficiency that are actually on par with that of the human brain, less than a femtojoule of energy per multiplied cumulative per synaptic operation here. So there's some great advances that could be made here then in neuromorphic engineering. So a few words on the counterpart of that. So in addition to doing neuromorphic engineering, where we take inspiration, in biology and in constructing silicon systems that implement it, we also have silicon interfacing with biology at scale to study and modulate a function. And so there, it just so happens that at each level of scale, we have this isomorphisms again. In this case, right, where, for instance, at the whole brain level, we have interfaces to the whole brain with electroencephalogram or functional near-infrared spectroscopy, right, that allows to probe this function. We have also other means for modulating a function at the whole scale, such as transcranial magnetic stimulation. And then down to all the level of molecules, we have molecular probes that we can also develop directly interfacing with silicon as well. And so in between, we have those arrays of electrodes that we can also, at this level of scale, we have this means for interfacing with biology and neurobiology at this level of scales, which now allow us to close the loop and actually have those intelligent, natural intelligence systems to interface with our brains at scale. And so we do this at extreme efficiency again, where at just 1 microwatt of power per recording channel, right, we can have great accuracy in just resolving bipotentials down to microvolts, because with microvolt root mean square input for noise, and this is actually a record of energy efficiency, of noise energy efficiency. And this allows to really then have large-scale monitoring of brain activity, such as here, where we did this with marmoset neuroscience recordings, right, at massive scale and at high accuracy. And so we extended this to recording from thousands, in this case, 1,000 of electrodes for lab-on-a-chip applications, where in addition to recording, we can do stimulation bidirectionally, either voltage recording and current stimulation, or current stimulation and voltage recording with the same circuit that is basically multiplexed in different fashions. And this way, again, we accomplish really great efficiencies. Again, 6 microvolt RMS at 25 kilohertz bandwidth, or 20 femtoamps resolution at 10 hertz bandwidth at just a microwatt of energy per channel here. So this really allows to really probe functions of the brain at scale. In addition, we can also then interface, in this case here, to the retina and through this nano-engineered retinal prosthesis. In this case, we even use silicon nanowires as, so at a scale that is comparable to the rods and cones that you have in the human retina, much better than today's typical Argus or so electrodes that are being developed for today's retinal prosthesis. So we can get much greater matching of the scale of our non-engineered retinal prosthesis at the scale of actually rods and cones, and have those nanowires actually being photosensitive, and then driven by electrical global electrical stimulation pulses that allow us to give us basically direct light modulation. So the impinging light through the eyeball onto this engineered retina will then stimulate locally those nanowires and drive them, make them electrically conducting. And then by a pulse, electrical pulse that is generated, just a single pulse, single scalar pulse, that we can activate basically the entire array according to the light that is activated. So we can get direct modulation of light and activated by electrical stimulation. And so this is much better than the status quo in retinal prosthesis, which either require an external electrode, an external camera that has to record the visual scene and then transmit this over a high bandwidth throughput wireless link to a local electrode array, where then this has to be multiplexed and basically the electrodes have to be stimulated in sequence. Or even if we integrate all this sensors that detect light locally, still we have some overhead that needs to be done. And so our system that is developed in conjunction and with a startup, nanosciences, biosciences, nanovision biosciences, a startup from UC San Diego, the Chicago School of Engineering, developed on this nanowire arrays that are much more efficient for interfacing than to the diseased retina for those impaired patients. And so we're able to actually record and also biopotentials as well, in addition to also record the amount of energy that's being transmitted, and this way allows us to actually precisely control the delivery of energy to this implant efficiently. So that's work in collaboration with people here also at the Center of Wearable Sensors, as well as wireless communications. And so we can close the loop here and have those sensors and actuators interfacing with the brain at massive scale, then also being used for remediation of neurological disorders. And here's an example of how this can even be done for Parkinson's disease remediation, even with totally unobtrusive sensors, unobtrusive neural interfaces, basically for sensing EEG through mobile brain imaging, as well as motion capture, and then closing the loop with force feedback for those Parkinson's patients, basically tactile sensing for that, right? And doing so in a way that by probing function of the brain and kinetics of the body, then coupling that to force feedback, we can actually directly bypass lost function of the brain and basically activate and drive the plasticity of the brain towards remediating disorders, in this case of Parkinson's disease, the tremor that we have used here. And so, and then we can couple that with models of neuromorphic engineering, that can guide this better closed loop neural prosthesis, in this case for remediation of this Parkinson's. And so there's several examples that we can give here, but so here we have this dry electrode arrays that interface to the scalp with then a direct brain imaging signal processing at the periphery that can actually decode location as well as function of these multiple sources of activity in the brain, owing to this signal processing pipelines for doing independent component analysis, basically implementing those neuromorphic pipelines for very efficient inference from the EEG data. So doing so, so this is a means for having real-time brain imaging, then couple them to close the loop for neuromodulation. And so one even more exciting avenue recently has been at the Center for Wearable Sensors, is that those sensors can be integrated even within the ear. So, and most people today, whether you're young and you're wearing those earbuds, right, with your iPhones, or you're elderly and you have this basically hearing aids, you have typically something in your ear that projects sound into your ear, and you might as well have a few sensors locally there in the ear canal that can actually sense your EEG signal quite reliably, and this way you can close the loop between sensing of brain activity and then auditory modulation of activity, with some really transformative health advances that can be done here. It just so happens that we can record not only EEG electroencephalogram, but also because auditory evoked potentials, even electrodermal activity, sweat in the ear, basically. And we have also demonstrated recently that we can record even lactate sensing in the ear. So those are all now possible. And coupling that now with activation in the ear gives rise to some really exciting avenues for neuromodulation for several diseases, tinnitus, insomnia, apnea, etc. So it's very exciting. So with that, I'll just, so we have some exciting things happening in Franklin Tony Hall, in the Center for Engineered Natural Intelligence, Center for Wearable Sensors, Center for Wireless Communications, and so we very much welcome interactions with our Japanese colleagues in industry and in academia. So we look forward to working with you. And so with that, I'll take a few questions if there's some time remaining.
Thank you, Dr. Cohenberg. There is a few questions from the audience. Let me read the question. The first question is, how can you overcome the natural brain in intelligence quality by silicon?
Oh, it's a very excellent question. And so we have seen some great advances lately in artificial intelligence. You know, those artificial intelligence, what is now called artificial intelligence, really is, I would say, neuromorphic engineering. It's really artificial neural networks. In fact, the founders of DeepMind, which now revolutionized this artificial intelligence in this latest, for instance, AlphaGo, as you have heard recently, right? The founders are actually neuroscientists, and they understand how the brain works. And so many of the advances that we see now in artificial intelligence are really inspired by, say, the layered organization of cortex, right, for deep learning, and then the basal ganglia, right, how basically the brain does this reward conditioning, right, of this temporal difference learning that we have, right? And so so naturally you can take all this inspiration and in building circuits that just emulate that. And so we have a very close resemblance. So yeah, our circuits are actually innovating the digital future, so that they inherit, they can get great, not only great efficiency in computing, but also they combine, as a mixed signal modalities, they combine both the efficiency of analog, but the precision of digital, because all communication is really digital with spikes, just the same way as the brain has done that. The brain is extremely efficient, but also extremely precise, even though elements are extremely inefficient and imprecise. All right, so the brain was able to do all that because you have this digital communication, the spikes that occurs in the brain. And so we use the same principles for doing efficient communication and precise communication, right? And preserving accuracy in our computation. And I'm not sure whether this I correctly interpreted the question, but so please iterate on the question if I didn't answer it correctly.
Thank you. So do you anticipate a number of, smart devices based on this advancement in the neuroscience? And
Yes.
Our human physical capabilities
Yes.
will be enhanced significantly thanks to the digital technology.
Yes, and the current brain-computer interfaces are still quite, I would say, even though you hear all those great advances such as Neuralink, right, came up with this assistance, but there's still, I would say, it will take a long while before we can really have implants that are quite useful. But there's some new advances now, and we also worked here at Franklin Antonio Hall in Jacobs College of Engineering. We have this large, we're building 4,096 electrode ECOG arrays. The Shadi Daya is leading the effort, and also part of that. Right? So ECOG stands for electrocorticogram recording, and that's minimally invasive. So you can imagine in the future, we would have minimally invasive surgery for placing those arrays of electrodes on the cortical surface. It doesn't penetrate the brain, so it's less obtrusive. And so you can imagine that we could have augmented cognitive capabilities for people that have their aging, where this can help. And so it is definitely possible to have the smarts that you have in neuromorphic engineering coupled with this ECOG arrays with thousands of electrodes to really project, stimulate as well as record and basically modulate and help to basically recover lost intelligence in aging or dementia, etc. Yeah. So there's some very exciting advances that lay ahead of us.
Thank you very much. Let's encourage our Japanese industry partners, academic partners to come visit us and see the collaborative work underway in your collaboratory. Thank you very much, Dr. Koenberg. So now we'll move on to the panel discussion.
皆様、今から数分かけてですね、パネルディスカッションの方の設定、場面設定に移りますので、少々オンラインの皆様お待ちいただければと思います。よろしくお願いいたします。
Now, we have a panel discussion about international acceleration programs for university-affiliated startups. UC San Diego has worked with several international partners to provide entrepreneurship and startup acceleration trainings. Today, we have invited Professor Hibara of Waseda Business School and Professor Koyanagi of the University of Tsukuba, as well as Mr. Abramsky of the UC San Diego Institute for the Global Entrepreneur. or IGE for short, to discuss the value of international programs for startups. First, let's ask our panelists to introduce themselves and very briefly talk about their programs. First, Professor Hibara, would you please start?
Okay, thank you so much, Waka-san. So it's really my pleasure to join this panel discussion. So I'm Nobu Hibara from Waseda Business School. So actually related to LINK-J, the supporter of this event. So even before the LINK-J was established 2016, I have been a supporter. So, I really, I'm really excited to see such a number of events happening in LINK-J right now. So today, I would like to introduce very briefly two programs. So one is Waseda Business School and UCSD kind of a collaboration. So every year, I take about 20 or 25 MBA students to UC San Diego every September. So that is the first one. And the second one is not related to San Diego, but Waseda University has been doing some, kind of a collaborative program with Israel. So that is about 3 weeks program, very intensive. So we did 3 times. So maybe, one participant in the Israel program will show up in the video later. All right, thank you so much, Waga-san.
Thank you, Hibara-sensei. Next, Professor Koyanagi, will you start? Please take the microphone.
Okay, thank you very much for having me here today. My name is Tomo Koyanagi from the University of Tsukuba. And first of all, congratulations for the launch of the new building and the facility. I'm very excited to listen to the new facility and also the project to combine the digital technology and many other type of the research area. So I personally from the Osaka University Engineering School, so I also very happy to listen to the project, a collaboration between the UCSD and the Osaka University. So we are running the project together with Jacobs Engineering School, School of Engineering, together with Professor Bremski. And we are sending every year 2 teams to that project. And we are working together with 6 universities from Japan. Today, I will introduce some successful cases of that project. Thank you very much.
Thank you, Koyanagi-sensei. Next, Dennis, will you please introduce yourself and describe your program, please?
Thank you. My name is Dennis Abremski. I'm very happy to be here today to talk about the collaboration that we're doing with universities here in Japan. I am the executive director at the Institute for the Global Entrepreneur. We are one of the only accelerators, university accelerators in Southern California that focuses on science, life science, bioengineering, and medical technologies. So we take the big ideas that are produced in the School of Engineering and transform them into something that could change the world because of the products and services that are created for the markets that we serve. Our startups have raised over $100 million here in the US, and many of our international teams are from Japan. So we're very proud of that. We've worked with groups like Research Studio, and our collaborations are creating startups that are translating research into the global marketplace. So today I'm very happy to be here and happy to talk about our collaborations and hopefully how we can grow them in the future.
Thank you, Dennis. we didn't have much time to really describe our programs. However, I just like to ask each of the panelists about the impact of the entrepreneurship and acceleration programs. Hibara-sensei, for example, have you observed any change in the teams before and after your programs?
Okay, thank you. So let me just share the maybe participant could feel the let's see, could feel the program. So that is a kind of a part of UCSD and our Waseda Business School program. So this is a kind of a study abroad program. So I'm responsible for this course. So called Financing for Life Science Businesses. So, we are visiting radio school management in UCSD, and some professors and some, practitioner talked about some initiative in San Diego, and particularly, the JJ Lab. It is a very, very popular to also business school student, so visiting that lab, so they could feel some real sense of what's going on in San Diego, right? So, and also Israel program, let me just share to show the taste of the program. So actually this program is more than 2 weeks long. So kind of a very good mixture of lecture and training. And also this program is not so focused on life science technology, but recently in Israel, in San Diego and even in Tokyo, kind of a convergence, right? So related to life science, AI and the space technology, every type of technology is very strongly connected to life science business. So in this program, XR, VR, right? And AI or even machine learning, and even blockchain. So Israel Scholar had a chance to talk about the detail of this type of new technology to participants in this program. So I observed really participant changed very, very substantially, right? Even very brief, 2 weeks or 3 weeks program. So, and particularly, they kind of feel how to get money, right? So actually, my research has been focused on venture capital and the entrepreneur ecosystem. So unfortunately, only in Japan, right? Only in Japan, students might feel that, too naive, particularly about money. So they got a very good sense of how to raise money in the international training. So actually, the video will show up. So he mentioned, right? He mentioned Tanaka-san mentioned some, experience in Israel program.
Okay. Then now let's ask to show the video, one of the trainee of his program.
Hello. Nice to meet you, everyone. My name is Hiroki Tanaka. I would like to introduce our business and Waseda program to you. I love architecture. I think people should have a space that is easy to spend and that allow us to be ourselves as we are in both real and virtual space. I want to realize it by my company. This is our team, specialist of architecture and programming from this company with support of Waseda Edge program. Now we work with 20 members. Our business makes architecture and space in virtual reality. This can create new jobs for architects in affordable VR space. People can sell products and services and work in a VR platform. They can gain information and make profit. We provide VR as a new location to enrich communication in real space. The top three diagrams display a function to increase communication and build relationship of trust. The bottom two diagrams are function for creating higher quality communication through analysis unique to online services. These are our customers. They enjoy having VR show up and get more profit by a product. Now I introduce what's the program for venture companies. I attended at No Venture, No Victory program in January, February, and March in this year. In this program, specialists in Israel taught me many things online. I could learn how to manage my company and get new knowledge of AI or blockchain. What impressed me most is in this program is Yossi and Tanya's session. In this session, I learned how to get the first customer. They taught us how to tell our services worth. I am not good at English, so this task is very hard. I try to explain my product in easy English words. It is a good opportunity for me to think about how to explain our product. After this program, I could talk with venture capitalists in foreign countries, and one of my customers is in Thailand. This program helped us to challenge to expand overseas. For further information, please visit our homepage and contact with any question you may have. Thank you for your time.
Okay, sounds great. Now let's ask Dennis. So can you talk about the change your programs have brought to the teams and the startups and examples?
Yes, the teams that we work with are typically researchers. Sometimes they are very early stage, but we work with them so that they understand their market and their customers' needs and how to tell their story. So similar to what you just saw, most early stage entrepreneurs are not very good at telling why they do what they do. They focus on the technology. So what we try to do is get them to understand the value they provide to the market and how they can tell that story, whether it's to a partner or to a venture capitalist. If they can get across the ideas that they're working on, they've done 80% of the work. The other 20% is to prove they can execute on the goals they set.
So let's show one of the videos that is fostered under Dennis' program.
So my name is Patrick Schmidle. I'm the CEO of Carry Therapeutics, and we're a company that's building an opioid treatment adherence solution. And I suspect that many of you on the call here know of someone that's been affected by this crisis. The good news is that there's hope, and that hope is a medication called buprenorphine. It's proven to reduce relapse rates and overdose rates, but the problem until now has been access. Only physicians with a special license were able to prescribe it until last month, which is when the current administration removed that requirement. So that takes care of the access. What we now have to solve is adherence. And the current alternatives for adherence, they just don't work for buprenorphine. They relapse, they end up in the ER, they end up in the hospital. And we estimate that the cost per patient that relapses is approximately $500 per month. So what's so groundbreaking about our solution is that for the first time, physicians will know if a patient has taken their medication, and if so, when and how much. So it's a closed loop solution that is a real-time confirmation when patients have taken their medication. And if you look at that little, the patch here, it looks like a Band-Aid for you, but if you turn it around, it has microscopic, tiny little needles that penetrate the skin and measure the medication that can then be transferred to the patient as well as the care team. And what makes us unique is that while other companies, they try to measure physiological biomarkers and physiological symptoms and various biomarkers, we focus on medication levels. And it's not just about measuring adherence, it's understanding if patients don't take their medication, we can ask them, well, why aren't you taking it? And then we can activate the app system to make sure that they continue with their treatment. So to the contrary, if you think about our solution, once a physician diagnoses a patient with an opioid use disorder, they can prescribe buprenorphine and the patch at the very same time. The patient can put the patch on, begin treatment immediately, and without having to go to the office and getting the dosing right, the physician can do that remotely. They can optimize the dosing remotely, and just that in of itself will increase adherence, because many patients are lost because the dose is either too high or too low. So there's a tremendous opportunity in front of us, and I hope you join us in fighting the opioid crisis. Thank you.
Okay. Now, Koyanagi Sensei, you have run the research studio program for several years. What is the impact of the program so far?
Okay, so probably it's easier to show my slide, something like this. So this is the cumulative number of the trainees, 74 trainees, and 74 trainees from 23 teams, and several of them were incorporated in between or after finishing our program. And fortunately, some of companies already raised more than $30 million so far. And 2 of them are getting to the clinical trial stage, which means that they are very close to the proof of concept of their own business model. And in our program, we are providing the training for to think about what kind of product they have to develop. So target product profile is one of the keywords. At the same time, we provide the training for the business model. In many cases, our acceleration program are focusing on either product development or business model. So everything has to be back and forth. We have to discuss a lot together. So this is one of the features, but at the end, we have to think seriously about the market. We are working on the life science field, and the biggest market is in the US. So we are sending the teams. So these are logos of our program, and logos with crowns. Our teams already went to the training by UC San Diego. So Dennis, you may remember the most of all teams so far. And for example, Vesica Intelligence, they will present after this as a video. They are having the meeting almost every week. So the CEO's English was getting better and better, I realized. But not only English, the business model is polished very, very well. And this is the actual image of our visiting San Diego. And I'm not getting into detail, but we got a lot of real experience to make an interview of the potential customer or potential partners. And fortunately, for example, like Dennis provided a chance for the startups to meet the KOL in the UCSD hospitals. So through these kind of experience, they already got a lot of sense of the real business through the training.
Yeah, so he has already described what has been done collaboratively. Maybe you can come up with a few more words about the collaboration and how you have managed, and also some of the success examples coming out of the collaboration.
Sure. One of the things that we worked on together with University of Tsukuba was to plot where we thought the future of their startups were going. Rather than make it a transaction between the two universities, we knew that it was a long-term commitment and a long-term relationship. So with Vesica, for instance, we understood what they could do because they were a software company. And in the United States, software is much more investable, but it has a timeline that lends itself to venture capital. The problem was, how do you get a venture capitalist in the United States to invest in a Japanese company? So what we did is we started a US company. We took the intellectual property from Vesica and got it licensed to the US company. But more importantly, what we did was we made sure that the US company will cross-license back all new research back to the Japanese entity. So what that means is now you have a multiply effect. The research grows because now you're connected to two global markets, and each of the entities benefit. And as the Japanese side benefits from knowing about the American side, same thing, San Diego knows more about what's going on in the Japanese market. Eventually, they'll move into other markets in Europe and they can do the same thing. So this is a model we're trying to use when it comes to life science, that the markets are so much different that rather than try to make old models work, we're working on new models to make it easy for these companies to advance.
Well, maybe it's time for us to see the video of the company.
Hello everyone, I am Atsushi Ikeda, Department of Urology, University of Tsukuba Hospital, and co-founder of the Vesica Corporation. We are a startup working on the digital transformation of the cystoscopy. Let me tell you about my friend Brad. He is a father and a husband. He was recently diagnosed with bladder cancer, but it was removed by his urologist. This brought great joy to his family. Today is his 3-monthly follow-up test. He knew that bladder cancer has a high recurrence rate, and up to 30% of bladder cancer are usually missed, but he's always been a lucky guy, so he was not worried. However, the follow-up test shows that his urologist found a new tumor, which means there was missed region in the previous surgery. Once a region is overlooked, it's usually not detected and developed into the bigger issue. Lack of detection and reporting have put Brad and his family into another downward spiral. As a urologist myself, I've seen cases like this many times. This has motivated us to come up with a cystoscopy support system, an AI detection system, and a data recording system that ensure that even these small and flat regions are not missed out. This is a video of cystoscopy. On the left is a video of conventional cystoscopy, and on the right is a video of real-time detection by AI. A highlighted region our system could quickly point out. Almost all regions in the bladder are accurately detected. Our technology also supports an image-enhanced technique, such as narrowband imaging. This case had a multiple papillary tumor on the left wall and a flat region on the top of the wall. The new recording system automatically creates a finding figure during procedure. This is a competition grid. We believe that our technology has an advantage over any competitive endoscopic technique like NBI and blue light. Cystoscopy market size in the United States, we have over 1 million cystoscopy per year. We estimate if our system usage fee is 10%, so our entry market is $66.8 million USD. We consider a business model where we charge per cystoscopy like a subscription. In the future, we will also provide the cloud-based service. This is our team. Our mission, unleashing the power of endoscopic medicine to open the future of patients and medical professionals. Thank you for your attention.
え 大学の先生とは思えないほどすごくビジネスっぽかったんですけども。So, 小柳先生, so you've mentioned that they have improved a lot over the course of the training. And so what made them improve so much in a short period of time?
Yeah, most important thing is the scientists usually think their scientific achievement can be translated into business very easily. So they said, oh, this is the course which someone could take his business into his technology into business. That was the initial their recognition. But through the course, they started to see, oh, maybe it's difficult for a business person to understand the technology in detail. And then he decided to translate his own research and technology into the business term. That's why he could present like business way. So all slides were very simple, and even the AI is catchy word, but AI itself is not only their strengths. So mapping system of the ball, you can see the bladder is not flat region. That's why it's difficult from the all other technology for the through the image recognition. So those kind of through those kind of things, and also he got a lot of training by the U.S. mentors, UC San Diego mentors. So through those discussions, of course, they have strong scientific background and also financial or like intellectual property. So by displaying the backgrounds, created much broader sense of the business and deeper discussion of his own plan.
Thank you. So Hibara Sensei, have you also observed similar tendencies among the team members after interacting with international mentors or advisors? They have realized something that is not necessarily readily available here?
Right. So, mentors, unfortunately, still in Japan, very limited number of good mentors, particularly business mentors, right? So we have to accept that reality. So therefore, maybe this the current Tsukuba case was very, very successful. But from my perspective, still very, very exceptional in Japanese university-based technology startups. So therefore, maybe it's if we could get good mentors, for example, from San Diego across Pacific Oceans, that would be very, very fantastic, right? But still we don't have kind of a recipe to get good members to Japan.
Can I add one comment? Yeah, I just want to mention that most of the mentor from the UC San Diego is serial entrepreneur. That's the biggest difference. It's pretty much difficult to find out in Japan. But we really thanks for those kind of opportunity to working together with San Diego.
Any comment about that?
Thank you. One of the things that we work with, reason we work with serial entrepreneurs is because they have sometimes failed. So they know what that means, and they try to advise the startups, and not just the CEO, but the entire team, that they have to work together towards a common goal. They have to prove to their teammates, they have to prove to their partners. So if you remember part of that video, there were examples of why it was important that Vesica was doing certain things. And it was very personalized. It was very customer-centric. So rather than say how you do it, you say why you're doing it. And that resonates with your partners, that resonates with investors. And it proves that you understand your market. And that's one of the first areas, the very first thing we do with a team is we ask them to tell their story. And then we deconstruct the story and say, how does your story connect to the same story that your customer wants to hear? Because they have the same needs. And once that deconstruction or that reconstruction happens, now we have them pitch to early but friendly venture capitalists. So it's not a pitch for money, it's a pitch for advice. We get them used to not asking for dollars, but always asking for advice, whether it's a partner, whether it's a teammate, whether it's a investor. And that's proven that we know how to virtualize what we do. So we hope to be able to do that with all of our new teams, but we would be happy to work with Waseda.
So we have discussed so far from the point of international acceleration programs and also the collaboration between institutions. And but it's not necessarily true for all the startups to go global. And indeed, startups are small, nimble, and also their financial resources are limited. So I just want to broaden the perspective a little bit here and discuss more broadly. So what can we do to really help startups expand business internationally? You know, the concept here was that, okay, UC San Diego IGE has been supporting the startups there, and how can we take advantage of that for the benefit of the Japanese university-affiliated startups? But that assumed that, you know, startups are encouraged to come to the US and operate like the US companies. But, you know, of course, not all the startups needs to go abroad immediately. So I just want to take this moment to discuss, so then more broadly, what can we do to help then expand business internationally, not necessarily copy the US startup models. Can we discuss that? So maybe Hibara Sensei, you have a comment?
Yeah, so simply, you know, the very quick answer to your question is what do we need is people and the money, right? So the people we already discussed, maybe good mentor from San Diego or from other countries. So that is one thing. And also the second thing is money. So I think that, you know, even in Japan, you know, good potential of money available to startups, you know, but still, you know, particularly technology-based, university-based startups still depend too much on the government money, I think. So LINK-J is a very good private initiative. So I hope that, you know, LINK-J also could be more involved with money matters in life science scene. But also universities also have to make more effort to prepare some good amount of money for startups in their own university. Yeah, for example, you know, my university, Waseda University, just established Waseda University Ventures, kind of a university-based venture capital forum, actually last month in April. So I really look forward to what will be going on in the Waseda University Ventures space. But maybe, you know, usually we are paying attention to venture capital activities to make startups well shaped. But my sense is that before we see financing, maybe gap fund or other type of money, in addition to research money provided to researcher in university, maybe that should be key point to make more startups happen in Japan. Yeah, for example, in Waseda University, actually 2 days ago, Denis and Waga-san came to Waseda campus and with me, and we had a good discussion with a couple of people of Waseda guys. So, you know, even in Waseda, so kind of gap money, 80%, 80% of gap money come only from the government fund. So therefore, you know, maybe, yeah, the things has been changing right now, but maybe university should get more their own money, right? Particularly for gap fund, kind of from endowment or something. So yeah, some university has just started to be headed to such type of activities. That should be more abundant. I hope in the near future.
Thank you. Koyanagi-sensei, how about what's your opinion of this?
Okay, yeah, the most, so actually on Tuesday, we had a session similar to this, and most of these venture capitals and startup people says we should take the fastest way to success. So either in Japan or US, we have to find out the best way, but it's depending on the definition of success. If we want to get into the market first, Japan origin company should develop in Japan. Maybe that will be easy, but eventually the product couldn't be sold in global market. It'll be difficult because of the difference of the regulation or other financial conditions. So we are providing the training. At least every single company should think globally, then decide where to develop their own business. So that's most important thing. For example, like a drug discovery company, they have option to license out to the global company from Japan. But if we want to get enough information of the customers, in those cases, big companies, big pharmaceutical companies, the company should be developed in Boston. But that is not the one single answer. And another case, for example, like a digital technology, which is focusing in focus of today's discussion, it will be a little different because the regulation is totally different. So product liability issues and also the FDA and PMDA regulations, these are totally different. That's why the Vesica Intelligence decided to get into the US first. For example, like ARM, Japanese startup, was acquired by M3 this week. And the size of the acquisition was like something like $250 million US dollars. Is that big or small? M3 is a domestic company. That's why it's depending on the strategy or mission of the company.
Dennis?
There's ways the universities can provide gap funding. One way is to develop university angel funds, where angels come in and see what's going on at the university level, and they can inject capital at an early stage, because most angels invest in something they know. So they become a knowledgeable investor, and they bring in the next level of investment. The other way is a studio model. And the studio model is to create a team around a core, either a core inventor or a team of inventors. And that's another area that we're looking at. So if a technology wants to come to the United States without a team, but it has a good patent strength, we can build a team around it and create a US entity. The reason for doing that is because the FDA regulations and so on are a lot easier to deal with. But the same thing could be done in Europe. The same thing could be done in Australia, right? So the reason sometimes it's done in the US versus other markets is the market need. So what we try to do with the startup is determine where they do the best, where they provide the best value in what market. And if that market is the UK or that market is somewhere else, then that's where they need to go, especially for clinical work. They can always come to the United States. They can always go to other markets. But time is the enemy with startups. Money is a translation of time. If they can't survive long enough to get to the clinical trial or whatever their milestone is, they won't survive. It doesn't matter why. So that's where we try to work with it. And hopefully, if there are more gap funds at the university level, that will definitely make our job easier.
I'd like to take this moment to ask if there are any questions from the audience. Because, the symposium today is to talk about this collaboratory. This is a new way of conducting research. And by having interdisciplinarity, multiple disciplines working together on shared problem, there will be increased productivity, research productivity. And the result could be translated into startups or companies or tech transfer to big companies. And we really need to accelerate the whole process, research and translation, through startups or through other mechanisms. And I hear that, during this time, the current administration in Japan is very eager to have more startups. We know that about 20 years ago, there are number of startups, university startups. But now we have to make sure that those startups will grow and be successful so that there will be a real impact on society. So without that, we are a little bit concerned about the future. So I'd like to hear if there is any questions from the audience, anything, that has been discussed during this panel discussion for that.
Okay, so hello everyone. I'm Al Pisano. I address this question to the whole panel. As you look out over the system that you have now, can you point out one major obstacle that would stop you from, one, working with more startups, two, accelerating those startups, and then three, constructing a pipeline to the technological source. So the speed of the startups, the number of the startups, and the source of the startups. Do you have any thoughts about what you must overcome in any one of those areas in order to move the program forward faster?
I want to know the meaning of the first question. So, you want to know?
So I'll explain a little bit more. If I think the panel feels that what they're doing is something very important. So how do you do more of it? In other words, how do you have more startups created so that your chances of success increase? The second part was, how do you enable them to move faster through the stages? That was the speed part. And then the third part was, what do you need in your organizations to grow so that you can handle this load? So that's the spirit of the question.
Okay, thank you very much. Let me go first. So first question, we are struggling with how we can get more teams to enroll to our program. So when I ask the same question to the many accelerators in the West, then they are talking about how they select a good team. But we are struggling with how we can accumulate or cheer up as scientists to get into our program. So that is the biggest challenge. But at the same time, it's related to the second and third questions. Kind of venture studio or creation model, we have kind of decided to provide the team or mentors, business mentors who could think business model for researchers. So kind of service as well, business model creation service or something like that. We have to think seriously because the condition is totally different in US and Japan. So for the acceleration and also pipeline, we are try to develop a good system to work with the UC San Diego, and we are calling that as under the name of the Japanese whiskey. So we try to grow something together with UC San Diego.
Okay, thank you. Very good question and a very kind of big question, I think. So, particularly related to number of startups. So I think that particularly Japanese life science researcher, university professor have to change their own mindset. So that is probably the most important. So, when I talk with US venture capitalists, so they often indicate that, current Japanese situation may be very similar to 30 years ago US situation. So particularly mindset of professors was very similar to US 30 years ago. And also, maybe I think the most important change needed is still university professor in Japan cannot good delegation, right, to their student, particularly to take care of the business originally from the laboratory technology. So that point, if that point could be changed somehow, I think a huge number of startup even from Japanese university.
One quick thought is a little bit about the environmental change. That faculty see the university as their partner. Similar to the university giving them access to funding or lab space, I think if a university can project that they are a partner and they are creating a partnership with the startup, and that it becomes a win-win for each of the two parties, that will help. Because one of the reasons sometimes that it's hard to find teams is because they go into stealth mode, where they work by themselves because they're afraid about the issues of intellectual property at the university. That's changing, and I see it as one area. The other areas that universities can help is that they do provide a version of a laboratory, an environment where teams can come together on campus and meet with faculty and other students so that they can get access to the resources that normally they have, it's very difficult for them to find.
Okay, thank you. I think we will continue to kind of keep exchanging ideas and actually program ideas and also do it together. We can share good practice or challenges in order to avoid those pitfalls so that we will have a strong partnership over the ocean and also see more successful companies, startups. Okay, so that will conclude the panel discussion. Thank you very much for the panelists, and would you please thank all of the panelists.
皆様いかがでしたか。今日はですね、朝から、ここまでパネルディスカッションまでやってまいりました。冒頭のプレゼンテーションしてくださった、トップサイエンティストの皆様、そして今のパネルディスカッションにご参加いただいたですね、皆様、本当にありがとうございます。 ご視聴になられた皆様もですね、いろいろと気づきを得られたんじゃないかなと、思っております。いかがでしたでしょうか。特にですね、フランクリン・アントニオ・ホールというですね、これデジタルフューチャーというビジョンのもとに作られた、素晴らしい、場所がございます。そこにはですね、ライフサイエンスの方のみならず、いろいろな領域、のクロスオーバーというかコンバージェンス、していくための、トップサイエンティストの皆様が集まっていらっしゃるということで、こういったハードの面、作るのはすごいことだなと。設計図上もですね、こう皆さんがこううまくこうつながるような入り口が、10メートルずつにしか、離れていないというような作りにしているといったところ、すごいなというふうに思いました。一方でですね、今日お話聞いてすごく感じたのは、ハードだけではこれはうまくいかないと。それに加えてですね、ソフトの面がすごくいろいろあるんだなと。今日ご登壇いただいたですね、ピサの学部長のストロングな、非常に強いリーダーシップのもとですね、強いサポート、豊富なサポートもありますし、ネットワーキングの仕組みとかですね、コラボレーションの仕組み、様々なこういったソフトの面、このハードとソフトのベストミックス、ベストコンビネーションがやっぱりこの成功の秘訣ではないかというふうにすごく感じました。特にですね、後半のディスカッションではですね、グローバル、あとアントレプレナー、その人たちをサポートするメンターのお話等々ございました。特にですね、日本、このリンク J できてから6年ちょっと経つんですが、随分変わってきてですね、いろんな、イノベーションをサポートする仕組み、国もはじめですね、大学、インダストリーでも起きております。で、これはどんどん進めていきたいんですけれども、やはり米国ももっともっと進んでいるところがございます。そういったところからですね、いろいろ学べたり、一緒にですね、コラボレーションできたらなと思ってます。そんな中で言うと、今日皆さんお感じになられたように UC サンディエゴ校、素晴らしいですね。トップサイエンティスト、そして先ほどの、取り組み、最先端の取り組みもございます。そして私日頃から感じてるんですが、非常にオープン、かつ、とてもフレンドリーな UC サンディエゴ校だと思っております。ぜひですね、皆様今日のこのシンポジウムをきっかけにですね、今日例えば来日されているピサの学部長、和賀さん、そしてデニスさんにですね、こうまさしくヘジテイトすることなくですね、コンタクトしていただいてですね、みんなでこのイノベーションをさらに進めていきたいと思いますので、よろしくお願いします。今日は本当にありがとうございました。